So let's move on. Now we're going to get into meiosis in a different video. For now I want to stick to mitosis because it is essentially the simple basis for our understanding of meiosis. So if we can understand mitosis, then meiosis is going to be pretty much the same thing with a couple little tweaks or twists. Okay? So the first thing that I want to point out is that there's a mnemonic for remembering, not even a mnemonic, it's like a word that you can use to remember the steps of mitosis. And so it's IPMAT, okay? And really, technically, if you really want just the steps of meiosis, or mitosis rather, it's PMAT. What the I is what happens before mitosis. So prior to mitosis, remember you have interphase. So the cell has grown up through gap 1. It has divided, or not divided, it has synthesized or created more DNA in the S phase, and then it has prepared for division in G2. Okay, so if you remember as IPMAT, it means interphase, and then we have the 4 steps of mitosis. So really these 4, the PMAT rather, these 4 are really mitosis. The I is what happens before mitosis. Okay, so this is before mitosis. Now let's get to the actual steps. So these 4 things right here, these are mitosis, broken down into 4 parts. We're going to start with prophase, okay? And so in prophase, you have your cell, all right? And you have, whoops, I'm going to clean this up a little bit just so we can have a little more space and we can see some detail. So again, we're talking about prophase. So there's our cell, and you have your nucleus, okay? And within that nucleus, you have your chromosomes. So let's say we have a cell with 2 chromosomes. Okay, so what you're going to see here is that the nucleus is going to start breaking up. Okay, so nucleus is going to start to dissolve, essentially, or break down. And then the other thing that you'll see during prophase is you'll start to see something called the spindle apparatus form. And so on each side of the spindle apparatus, you'll have something called a centriole. Okay, and then in between the centrioles, you will have the spindles, or spindle fibers, which kind of look like threads. Okay, so just to be clear again, nucleus is breaking, nucleus breaking down, and then you have your spindle apparatus forming. And these little guys at the end, which are essentially the spindles, you can think of these, or these are called the centrioles. Now, the question I usually get is, okay, that's the spindle apparatus, and these are the centrioles, but what the hell is a spindle? Okay, so the best analogy I can think of is if you're aware of how fishing works, or a fishing pole works, right? So on a fishing pole, you have the reel, right, which spins. So if you had a reel like so, right? And you have your fishing line on there, right? And then you have your, your string, and that goes, right? You have your little like handles, okay? And then here's our fishing rod, okay? Really bad fishing rod. But anyways, so there's our line, okay? And our line is running along the fishing pole, okay? So think of this as the centriole, all right? And the centriole can cause these fibers or strands in between the 2 centrioles to tighten or lengthen. Just like how you can spin this to reel in your line, or you can let it out to give it some slack. So the spindle, or the reel, has the ability to manipulate, tighten, or give slack to release the fibers in between. Okay, and we'll see that these fibers will have some function similar to a fishing pole in terms of reeling something in or letting something out. Okay, so what we've really done in prophase is we've broken down the nucleus, allowing the chromosomes to float free in the cytoplasm, and we've created something called the spindle apparatus. Okay, so now let's move on to part 2. Okay, so we've talked about interphase, talked about prophase, now we're going to talk about metaphase. Okay, so with metaphase, you still have your cell, okay, but what has happened is our spindle apparatus has moved so that the two centrioles are on either side of the cell. They're on opposite poles of the cell. Our spindle fibers, okay, are running the length of the cell, like so, in between them. And then attached to those spindle fibers are your chromosomes. So somewhere towards the end of prophase, your chromosomes will get attached to the actual spindle fibers, okay? And then as the spindle apparatus slides into place, it will pull the chromosomes to the middle. And this is how I remember what metaphase is all about. The M in metaphase, I think of it as standing for middle or midline, and what that means is that the chromosomes have been pulled to the midline of the cell. Notice they're sitting kind of right on the equator, or the midline of the cell. That's how you know the cell has reached metaphase. So prophase, we broke down the nucleus, spindle apparatus started forming and pulling the chromosomes around, and then metaphase, it has finally pulled the chromosomes to the midline. Now anaphase, okay? Ana, think of this as meaning opposites. Okay, so right now our chromosomes are sitting right at the midline. Okay, and in order to understand what happens in anaphase, you have to remember what a chromosome looks like. A chromosome, right, is made up of two chromatids, right? They contain identical information, meaning all the genes in here are also on this side. They're basically like two different DNA molecules that are identical to each other in code, but they're just traveling around with each other, right? And then in between the two sister chromatids, or attaching them together, is our little centromere, right? Don't confuse centromere with centrioles. Centrioles are on the spindle. Centromere is holding the two chromatids together. So what can happen is if the centromere breaks, you can essentially split your chromosome. You can have one sister chromatid and another sister chromatid, and the centromere is broken. Okay, so we understand the chromosome. Okay, two chromatids held together by a centromere. You can split it into two sister chromatids. Right now, this spindle apparatus has tugged these chromosomes to the midline. Okay, and you can think of it as a little tug-of-war. This line is being pulled in by this spindle, and this line is being pulled in by this spindle. Same thing over here. This is tugging in this direction, and then on this side it's tugging in this direction. So at a certain point, when anaphase kicks in, your chromosomes will split. So if I redraw it, during anaphase, okay, you have your cell, you have your centriole over there, and you have your centriole over here, okay, and you have your spindle fibers, right? You'll see why I drew those short in a second. Okay, and then you have your chromosomes. Only our chromosomes have now been split. We've taken the chromosome and we've split the centromere, the part that was holding the two chromatids together, so that now one chromatid is going this way and the other half chromatid is going this way. Okay, and so ana again means opposite. So you're splitting your chromosome and you're sending half of it this way and half of it that way. All right, now in our final step in telophase or telophase, however you want to pronounce it, the chromatids are going to continue their journey to opposite ends. Okay, the spindle apparatus is going to come apart or start to dismantle, and our nucleus is going to start to reform. So you can think of telophase as being the opposite of prophase. Okay, it's the opposite of prophase. So if I were to draw this out, I would take draw my cell again. Okay, our spindle apparatus, whoops, draw my cell again. Our spindle apparatus again, our little centrioles, okay, they're going to move off to the side. The spindle fibers are going to go off to the side. They're going to start to be dismantled by the cell and put away until it needs to divide again, okay, and then we are going to get basically our chromatids, right, which are now going to be essentially the chromosomes of a new cell over here, and our other chromatids over here, and then our nucleus is going to start to reform. but we're going to get 2 of them. That's why we had to move the chromatids off to opposite ends of the cell, so that these can become the new nucleus for one of our new cells, and these can be the new nucleus for the other cell. Okay, so this is telophase, opposite of prophase. Our spindle apparatus is dismantling, our new nuclei are here, and then the final step is actually different from mitosis. Okay, at this point, okay, once we finish these 4 things, mitosis is over. Following telophase, or really kind of towards the end of telophase, we'll go through one more step called cytokinesis, which is the literal division of the cell. So what you start seeing is, you start seeing kind of a stretching at the middle of this cell, sometimes referred to as the cleavage furrow, meaning that this part of the cell will kind of pinch in and start to get really thin. Okay, even the walls, or not the walls, but the cell membrane over here will start to kind of like pucker in like that. Okay, and it will continue to pucker in and pucker in and pucker in and pucker in until it finally just splits right down the middle. And now we have a cell here and a cell here. 2 little baby cells, each with their own nucleus, each with their own chromosomes. But the key is we had to go through mitosis first so that we could split the nucleus into 2 nuclei, and then we could split the cell. If we did cytokinesis first, then one cell would have a nucleus and the other one wouldn't. Alright, so now once we have these 2 new cells, they'll go off, they'll go through interphase again, they'll grow to full size. When they're ready to replicate again, they'll go through S phase, G2 phase, and then mitosis all over again. So the order is PMAT. Okay, meiosis itself is PMAT, but interphase has to happen first. It has to grow up, it has to copy its DNA, and it has to get ready for division. Okay, make sure that you complete the questions online, the WISC questions, and we'll see you tomorrow.